Sorry for this. Sorry.
Okay, got right. Hives less.
And ok, yes, non. Ah, qu'est-ce que j'ai fait? C'est bon. Sorry. Should be all right now.
Sorry. No problem. I'll go back a slide. So we've talked about consumption. But the fact that prey are not sitting around and can respond is this whole body of literature that's non-consumptive effects of predators on. Now, that means predators can regulate prey populations in two ways, right? Through these consumptive effects, which we understand in classic ecology, and through these fear effects. And there's a lot of vocabulary associated with each of these pathways. You see them redundantly being used when sometimes they shouldn't be redundant, but they're all talking about the same thing. To the consumptive or density-mediated interactions are more direct than the non-consumptive effects that are driven by fear. And these are also called risk effects. So risk of predation changes the prey phenotype, and it changes all of this. It can change phys, which we expect, your stress response increasing and heart rate increasing, that's basic. Immediate response to predators. It can change morphology, behavior, life history. But for predation risk to have ecological consequences, which means going all the way to prey population size, there are a bunch of arrows and pathways that we have to be super explicit about, because you can't jump from here to there. So there's a chain of events that links predation risk to prey populations. One is right here at the individual response, right? The prey phenotype changes in response to risk of predation, not being eaten, because you're eaten, you're done. That change in phenotype should affect fitness metrics, like fertility, survival, or growth, which then influence population growth rate. Those fitness metrics finally result in a population size change. So being explicit about this chain of events allows us to see when it works and when it doesn't work, or when it breaks and when it pulls. Now, each of these arrows is a transition, and so towards the end of my talk, I'll come back to each of this transition, but this is the framework we want to work. Yeah.
So when you say changes in phenotype, you mean like heritable genetic changes or plastic response?
Plastic right now.
Okay.
I'm talking about plastic right now. At the individual ecological timescale.
Okay.
Evolution will touch on that at the end. But right now, inducible.
Okay. Thank you.
Yeah. Okay, so how do we measure these risk effects? Right? There have been several approaches. And not predation effects, risk effects, fear-driven effects. Right? We can experimentally manipulate the intensity of predation risk, both in the absence of consumption and in the presence. Right? So we can have predators be there, but not allow them to eat, and just scare the bejesus out of prey. Or we can take detailed and long-term measures of predator-prey traits and density under natural conditions. As you can see, the first one is easier to do, manipulate. The second one, we really have poor data for. But I want to say that with the world is changing around us, we might actually be able to capture number two more often than we did before. That's kind of the message that James Essex's original paper was. When we remove top predators from the environment, what happens? And anthropogenic change has become a rather convenient tool for us as ecologists. So it allows us to look at the outcome of both consumptive and non-consumptive effects. So I'll take you first to one study that we did to try and disentangle consumptive and non-consumptive effects of predators. And I did this work with students of mine at the oldest running wind farm in India. It's up on the top of these plateaus in the Western Ghats. And we asked the question that James asked 8 years ago, which is what happens when you lose predators? And so this plateau is not very long. which is 27 kilometers, and in it we found sites with and without wind turbine. So an anthropogenic change, and we picked that one. And you look at it here. This is the landscape. This is peak summer when the prey that we're interested in is out and about. It's simple. It's a simple ecosystem. We don't have a forest with lots of connected pathways. It's a simple ecosystem that has 4 levels. The wind turbine level, which is our creation on it, but it has raptors and lizards and insects. I'm going to focus on the birds and the lizards today. So if wind turbines come in, and we know this from literature for a while now, birds get affected first, right? They're not just like colliding anymore, they're just avoiding that space. So as a consequence of having wind turbines in the landscape, the first thing we'd expect is the apex predator in the natural food chain is going to reduce in number or reduce in activity. And we asked if that was true. So does the presence of wind turbines result in numerical changes at different trophic levels, right? Numerical in terms of density. And it does, but the apex predator densities and activities are lower in the wind turbine. So those are the red box plots there. So predator bird abundance, which is our raptors, were like half the number in places with wind turbines than in places without. And how often they were attacking was also reduced. Now, if the consumptive effect of like trophic cascade does cascade down one level, then we would expect prey densities to respond accordingly. Right? So where we have wind turbines, we don't have birds and we have lots of lizards. So already we see this numerical change that is predator driven caused by something that we did in the landscape. Now, beyond the consumptive effects, not wind turbines induce predator release, right? And the predator release affected the phenotype of prey. Right? And so we saw a whole bunch of non-consumptive effects. So the lizards that survived on these landscapes were different. So lack of predation pressure changed the phenotype in several ways. One, it lowered everybody's stress responses. So again, in red here is the wind turbine sites, and you see 2 bars, and the blue is non-wind turbine. The smaller, darker bars are baseline corticosterone levels, taken from with blood that we took from lizards out in the landscape. And the lighter bars is stress-induced. So we wanted to see how much can they mount a response to stress. And overall, cort levels are lower. Their anti-predator responses are also lower. They were super bold. And this is a classic assay to measure anti-predator responses. How close can I get to the lizard before it runs? Huh? And I can get really close. It got to a point where we were like, get out of the way. Right? That's crazy. Crazy lack of anti-predator responses in a predator-free world. Huh? It also lowers body condition. Now, and then it does things that affect their sex lives. So here we are, we were talking about all these survival traits, but the lack of predators lowered the intensity of sexual coloration. They became less bright and less like saturated. And that is the blue and orange patch on their dewlap. Lack of predators increased their aggression. So when we put males out onto the landscape, right, and gave them intruders to fight with, they would fight. But in areas in the absence of predators, they would fight. So they were changing their behaviors in ways that had nothing to do with survival, right? It's affecting axes that are related to social sexual outcomes, reproductive outcomes. So where does this take us? Right? What the wind turbine has done is remove the apex predator as a main player in the food chain. And that changed the whole suite of things for the lizard. Right? It increased density, that's our numerical effect. It increased boldness, lowered stress levels, lowered body condition, created males with dewlaps that were paler and duller, and they were more aggressive. Now, this whole suite of phenotypic traits, we had to sit and think about now, is predators or the absence of predators, can it really explain all of this? Is this a whole set of phenotypic changes caused by just the absence of predators, and it wasn't a very satisfying explanation. And in fact, what I think was happening is twofold. The predator release allowed the top stuff, the density, of course, right? But changed things related to survival. But it's because of that higher density that has now increased competition, a whole another set of traits has shifted. So while studying predator effects, we discovered competition as a main player in the phenotypic change. Okay. So this means if I'm trying to link that to a food chain, right? The wind turbine situation resulted in fewer predator birds, and it actually meant that we created a new food web where the wind farms are the new apex predator, right? Because it's taken the lower level away from it. So now wind farms are the new apex predator. It's resulted in fewer birds. It's resulted in more lizards and changed. So we're now playing at that orange level, like orange bubble, at that level. A little sidebar. Does it really matter if the phenotypes change, right? So I can measure it because we're scientists, we can measure anything we want and as much detail as we like, but does it matter? Is really the question, right? So do perceivable changes in phenotypes matter? Now, just as a sidebar, of course it does, right? And there's something as important as color, which for Sarda superba, they can see it. They're producing these highly reflective colors on their dewlaps, and visual modeling tells us that they can see these colors. Behavioral experiments where we show them dewlaps of different colors tell us that both sexes can detect these colors. Now, if the quality of these colors change, it could actually affect reproductive consequences. And in fact, the robotic kind of experiments we did, and I'll go back a second, were manipulated in several ways, where we created little robots that either had the natural colors or they had like one of the colors, right? Either all blue, which is unnatural, or all gray, or black, which is unnatural, all orange. And when doing experiments like this, where we give them different dewlap colors to see, we realized that not only can they see it, they care very deeply about which colors they're interested in. So, for example, sexes have different color preferences in this species. Males prefer the blue and black. So when given colors to see, they will watch blue and black for a lot longer. It draws their attention and it keeps their attention. Females, on the other hand, really couldn't be bothered about blue and black, but orange was interesting to them. So now again, if we go back to that idea, if predators are shifting, or now predators changing competition, which shifts colors, what does that mean for them? Again, as a sidebar, how you make these colors gonna matter, right? Blue is structural, black is melanin, orange is a whole cocktail of carotenoids and terpenes. Now, if you remember from broader literature, you've got your eat your way to orange. So now that might be the mechanism by which high competition in predator-free places reduced access to good orange-making food. And that could explain why some of our males mechanistically were not as intensely colored with high competition. So, intraspecific competition matters. And we see that in our interline size. But what about interspecific competition? Now, in that food web I showed you out there was nobody else in that landscape. They were the only lizard, and the plateau was theirs. But we have other closely related species that do live together. So Sarda darwini, which is a different species from Sarda superba, who you just met, lives with Sitana laticeps. And they both live in allopatry and in sympatry. You can see they're very different looking lizards. Darwini is gorgeous and tri-colored dewlap. Laticeps is gorgeous and it's all white. But and we're talking about like teeny tiny lizards here. So if you look at the scale, like this is what they're tiny. And what I show you here is the amount of color in color space that each of those lizards are producing. And these are males of the species. So what about interspecific competition? So this species lives in a different part of the Western Ghat Mountains. Right? I didn't really orient you to the map of India, but that's the map of India. In the western side of it, we have this mountain range called the Western Ghats. On one plateau is Sardar Superbug. He doesn't live anywhere else except there. On a different section of it, we have Laticeps and Darwinii. And they live apart as well as in one zone that overlaps. So now what is happening when you've got competition interspecifically? So the first thing I want you to notice is, I mean, this is a big graph, so we can let's ignore that left side of it and just look at panel C. Panel C is the description of how big their dewlap is relative to how big their body is. In other words, how much have they invested in that sexual signal? And the dewlap flaps. So the bigger the signal, the more obvious it is. And what you see on the x-axis is Laticeps on its own, the white species on its own, and the whites together. And Darwinii on its own and together with Laticeps. Laticeps changed in sympatry. Their dewlaps are a lot smaller relative to their body size. So interspecific competition has changed the white dewlapped male to invest less in his signal than if he was alone. It doesn't affect the dominant colorful lizard, right? Behaviorally, it does the same thing. Again, I want to just put the arrow so you can see the shift. Laticeps his flapping rate also reduces in sympatry with a dominant colorful competitor. Now, this is important because females of the species look almost identical. Right? It took us ages to tell them apart. And it's a tiny little scale difference on their dorsal that we finally figured out who is who. So for a male who's flapping his dewlap to attract females, right? He's discriminately flapping, and he does. He flaps to everybody and all competitors, right? The female then makes a decision whether he wants to make with her or not. And so in sympatry, the risk of getting the crap beat out of you if your Laticeps is high because the colorful lizard is bigger and more dominant and will take you down. And so in sympatry, competition with a different species can change sexual signal. And so when I was talking about challenges in the beginning, we said predation and competition, it can extend to this. All right. So we've been going along this chain, and we're still at the orange. And now I want to add a different selection pressure that we discovered along the way as being super important. Right? So it's not just predation risk affecting prey phenotype, but it's also competition. And I've also been talking about behavior, morphology, and physiology, but not mentioned life history. So let's take a moment to think about life history, because life history does affect whether the next chain in this event happens or not. And this time I'm going to take a much broader scale, an ecoevolutionary scale, and think about lizards all over. I'm not just pantherers in the Western Ghats. And I want to remind us all about some broad patterns that we see, and one is latitudinal gradient. And we see latitudinal gradients in phenotypic traits for every taxa that we've looked, in fact. So the latitude gradients are driven by these common abiotic components, right? The climate, temperature, seasonality, and all of that affects species richness, species abundance, metabolic rate, body size, life history traits. We're going to play with that bottom life history traits. For octotherms, literature tells us that a bunch of life history traits change with latitude. So as we go from equator to pole, age at maturity increases, longevity increases, clutch size decreases, clutch frequency decreases, but hatchling mass increases. So this is all ectotherms. But we don't build our bodies in the same way. Unless we think about the things that go into building the body. So I'll come back to this photo. How does a lizard build its body? It's got to eat the right things, enough of it to invest into different components of its body. So shouldn't diet influence life history traits and fitness outcomes? And if we look at the latitudinal gradient and be super explicit, oh, I lost the, on the left side was the map that has the gradient. Okay, so we want to, so if we're thinking about latitudinal gradients and traits, why are we not thinking about it with a mechanistic explanation in there? And one mechanism is food. So is there a role of diet in latitudinal gradient? So what is food, right? We can break dietary processes into these three groups, right? Carnivores that eat protein-rich food, herbivores that eat less nutritious, more difficult to digest plant food, right? And omnivores have the luxury of both. Now, it's to be a herbivore means you actually have a lot of challenges that carnivores don't have. They have longer gut passage times. They have lower assimilation efficiency for the same amount of food that a carnivore is eating. Its total energetic requirement is higher than a carnivore of the same size. And so as a consequence, a herbivore needs to eat more than a carnivore at any given point in time just to get to the same nutritional level. Now, add that to the fact that there's a latitudinal gradient in temperature, right? So herbivores at higher latitudes may be challenged with obtaining sufficient feeding time to compensate, right? Because it's cold and you don't have that much time to eat. And so this is what we predicted would have consequences for life history processes and performance. So this is our hypothesis here. So we went into this global study thinking that the latitudinal gradients that we see broadly, when split up by diet type, will give us different slopes, right? And that herbivores are going to suffer in terms of life history traits at colder temperatures than carnivores. So we curated data for 563 species that had diet info that we could classify as carnivore, omnivore, and herbivore. And that's the colors that are there. Carnivores are orange, herbivores are green, omnivores are purple. But it also needed to have all the life history traits that we're interested in. And so this is our data set. And the first thing you see here is actually the slopes of the lines change depending on what diet you have. So we see latitudinal trends in survival-related history, life history traits shift. So carnivores and omnivores, which is purple and orange, increase their age at maturity as they go from equator to the temperate. So they mature later in the temperate. They live longer in the temperate. And their reproductive lifespan, how many years they can produce offspring, is also higher in colder temperatures. But those green lines are the herbivores, and it's in the opposite direction. Right? So they are suffering. Herbivores have their age at maturity doesn't change, but their longevity decreases and their reproductive lifespan. So actually, not all lizards are the same. Right? It actually does matter which lizard, what you're eating to fuel your body. We have similar slope changes when we think about reproduction-related traits. And I'll just summarize here in a table so that we can see it all together. That our carnivores, and this is gradients, not absolute, right? So as it gets colder, their age at maturity, longevity, reproductive lifespan increases, but the number of clutches they produce and their hatchling mass are smaller. Herbivores suffer in a lot of ways. Their longevity decreases as it gets colder, reproductive lifespan decreases, and hatchling mass decreases. Now, if I was going to force life history theory in here, I would tend to call the carnivore body in a gradient as a slower life history strategy as it gets colder and colder, and the herbivore could be considered a faster life history strategy. But nonetheless, all these traits are shifting, but what does it mean ultimately for fitness? So we calculated one measure of this, which is lifetime reproductive output. Okay? So this is how many babies, how often, how big are those babies, and for how long have you produced them, divided by your female mass. So it's per gram female, what is your reproductive output? It's a matter of fitness. Don't forget, it didn't matter if you were carnivore or herbivore or omnivore, everybody had more or less the same lifetime reproductive, which is super interesting that the latitudinal gradients in this fitness metric, which is lifetime reproductive output, doesn't differ with lizards of different types. And this is because of all the trade-offs that are inherent in here. The lack of a reduction in reproductive output per unit female is because some things were increasing, others were decreasing, and the net outcome is that everyone's okay. So you could eat different things, even like this, protein-rich diet versus low nutrition, low digestion efficiency, but the net outcomes. So you can build the same body in different ways. Okay, so we've now we're here, and I took the life history corner to an eco-evolutionary level. We can also think about it at the inducible level, but nonetheless, how do we continue this rationale, right? How do we now say if phenotype affects growth rate, if growth rate affects population size? Because as ecologists, the overall goal is to see if fear itself can change ecosystems. And there's no point thinking about ecosystems changing if we don't expect population size and demography to change. So the question I want to ask here, if predation risk is chronic or very intense, what will happen to prey? In other words, can prey be scared to death? Can you lose them entirely? Now, lots of very important people have said that they can. So Evan Pricer says trait-mediated effects, the non-consumptive effects, are generally strong or stronger than the effects of direct consumption. It's a very big claim. Gary Blonsky says that top-down controls of population and food webs are enhanced by prey trait changes with predation. And this effect may exceed predator mortality itself. Again, big balls that fear itself can cause these dramatic changes in prey. And so in that first transition, the transition I've already showed you with my empirical work, it's been supported forever by ecologists and physiologists and behavioral ecologists for ages, right? So risk can induce trait changes in prey. I've seen it, so have many people. And they do all sorts of things, right? We've got behavioral hypotheses associated with that, like risk allocation hypothesis or food trade-off hypothesis. You've got defensive structures that Daphne are really famous of, right? They produce those big spines when a predator is in the water body with them. You've got physiological changes like stress hormones, metabolic rate, and you've got life history changes. And we've got lots and lots of evidence of transition work, not just from my own work. Transition 2. These trait changes influencing prey. I showed it from my work, it does, but others have shown as well that, growth rate can change via reduced foraging rate. So predator reduces your foraging rate, which has implications for how much you can invest in reproduction and survival, and so therefore you grow less. Fecundity, via reduced foraging again, or elevated stress, right? Too much stress because of the predator, and you don't produce as many babies. Probability of mortality, get via stress phase or poor condition or increased susceptibility to parasites and other predators. Right? These are the mechanisms by which transition 2 works. Transition 3 is the most difficult. Right? Can these risk-induced changes in fitness actually affect prey population abundance? Right? And this is the title, that Scared to Death Phase Was Not Mine. It was Evan Price's. Right? In 2005, he wrote this massive review that said that the effects of intimidation and consumption of predator-prey interactions, and his punchline was that the non-consumptive effects can change prey abundance. And so we did a deep dive, my colleagues and I, and we wanted to see if the evidence is really there. Right? Pricer says yes, risk can change prey. Right? We think that this is exactly where that chain breaks. And so our literature survey spanned a long period of time, and we found a lot of records of people measuring predator-prey interactions in various ways. And we excluded a bunch of things. We excluded experiments that did not allow changes at population size because they were manipulating individuals. We wanted to let those individuals sort of get to the end without manipulation. So we have to exclude those kind of experiments. We have to exclude experiments where they didn't allow recruitment to occur. And we have to exclude experiments where they manipulated the population size. The whole point was to measure population. And we found two categories of studies. Right, and so this is a paper that we published in 2020 in Animal Ecology. We found studies that compared prey abundance in two conditions, where predation risk was there versus predation risk wasn't. And then we found another set of studies where prey were exposed to risk and they allowed them to induce a response. So that first step, prey have to do something. And then they compared predator consumption of prey from those that responded versus those that didn't. These two category experiments, categories of studies actually measure two different things. The first one measured the cost of risk-induced changes, and the second measures the benefit of responding. So these are like not explicitly described as such, but depending on the experiment, we're actually measuring what happens if you're exposed to predators, do you die or not? Or if you got to fix it, right, did that help? Right, so that's the benefit. And in that 14,000 literature, we found very limited evidence of people who followed the entire chain. And in fact, only 8 studies fall here and 3 studies fall here, which means we're in dire need for real, proper empirical data to support this chain. And yet we see papers constantly say predation risk is an important selection pressure and it affects prey and have consequences for population density in communities. I say that in my introduction. I used to, at least until we did this review, because now I can't say that because we actually do not have the evidence. And the reason we don't is because we expect two things. We're a bit muddled in how we think about predation. We expect predation risk to reduce prey density, right? Because they're scared to death, right? But if prey respond, they induce a trait change, right? Build that cast, have a stress response that allows them to run away. Then that should actually give them an advantage. But if it gives them an advantage, they should survive that risk. So we have two pathways with two opposite conclusions. That risk should either kill you or not kill you. So the problem is that very few experiments separate those two outcomes. Very few experiments do the relative benefit of cost of responding or not. It's just really poorly designed studies. And we're not really thinking about going all the way here to think about what happens when you do or do not respond. And a lot of this comes down to things like this, really good excuses. I call them excuses because I think we can do better. Everyone says, oh, they're species and context specific. Right? Of the studies that I showed you, the 8 and 3 studies, all sorts of outcomes happen. So here I say that you could have reduced prey density in the response to predation risk. Some studies showed that. Other studies showed no change or an increase in prey density. So all possible outcomes existed. And it's because of these, right? People say species and content specific matters. The temporal dynamics are nonlinear. So prey suffer first in some experiments, and then those that can produce an anti-predator response that works, they survive. And then after a while, density goes from low because the bad ones die, and the fitter ones increase later. So it depends on how long you did your experiment. If you stopped it too early, predation risk will kill you, prey. If you allowed it to go long enough, predation risk may allow prey to recover, and you end up with a phenotype that is more successful. So some of our problems is that we haven't waited long enough. Or we have communities that are either super clean, where it's one prey, one predator, or super complex, a lot of competitors in there. And when you add competitors, it's hard to disentangle competition and predation. So can predators influence prey population abundance through these two pathways from here? No problem. Right? Of course, predators can eat prey down. So the blue arrow is no issue. But I think for us to be able to say that predation risk itself can change communities by changing prey, we need to bring consumption back into these predation risk studies. We need to actually do this, which is measure prey responses in predation risk in the presence and the absence of consumption. Only then can we boldly say in our introductions and our discussions that risk itself is as bad as being eaten. Okay, I'm done with my story here, and I just want to bring us back to this pretty lizard, was the focus at the very beginning. And the fact that for us to understand the phenotype here, we have to actually think about all these type scales. We have to think about how it's responding at the individual level, right? To challenges that could be competitors or predators, at the ecological time scale, as well as the evolutionary time scale. And with that, I'm done. Thank you. Yes.
Coming back to your experiment where you have I would like this
Yeah.
situation with your two visits.
Yeah.
So you had a kind of a reproductive character displacement.
Yes, we do have, yeah, exactly.
In the white species, right?
Agonistic character
And so you
displacement.
mentioned that the group of species was larger and more dominant.
Yeah.
Is there a difference in terms of abundances of those two species? Because my expectation would be that selection for reproductive character displacement would be higher in the rarer species.
Yes.
With the lower abundant species.
Yes.
Because they are more likely to encounter female from the other species.
Yeah, yeah.
So it's
There wasn't. Densities were almost even.
okay.
So
you interpret the fact that you had reproductive character displacement in one out of the two species because of the differences in terms of dominance and size between.
that's right. That's right. Because if you look at them on the plateau area, like it's a strand, after everybody's sort of all muddled up in there. You know, the males are defending like little, right? They're defending tiny little rocks. And so anyone can see everyone. And so there's a lot of fighting. And there's a lot of attempted courtship to their own species and the wrong species. It's really messy for them. And so the encounter rates of both conspecifics and heterospecific is super high. It's just the cost to the smallest species when they have a heterospecific encounter is really high. And that's our explanation, like our expectation of the agonistic character displacement, not the reproductive character displacement. The females don't. They're not the ones who's driving the character displacement. It's the fight. Mhm. Yes.
Did the wind farm implement any conservation strategies in relation to the results of the study?
No, yes and no. We did talk to a lot of the wind turbine companies about being more sort of careful about where they choose to place their wind turbines. But this is an economic debate, right? And not just an ecological debate. So there were no consequences in terms of them moving out of that plateau. Everybody now knows, because of the study and all the press that came with it, that this species lives nowhere else. Cerda subulata is found on one single plateau in the Western Ghats. And so it's become a tourist destination. And so it's in the wind turbine company advantage to not muck it up so badly that we lose this one species, and so they're not mucking it up. They're just not putting more turbines. What turbines they had that they had.
Questions. So one last one.
Yes.
So you said, because it's new to me, this idea of peer-induced
Yeah, non-consumptive effects.
effects.
Mhm.
But I'm wondering whether, I mean, when you say so you have effects that are directly linked to consumption
Yes.
that impact the prey population sizes, right?
Yes.
This is what induces, for instance, changes in the intensity of competition.
Yes.
So it's not directly, so the increased competition, you would call it, has a direct consequences of the consumption
That's fine.
Yes,
I'm not sharing this. Right?
yes, the consumption. Yeah. I mean, the evidence of non-consumptive effects, the fear effects on prey phenotype and prey fitness is actually a lot. There really is. I could give you like a lot of really interesting examples. People have studied birds where they've done playback calls of predators, and then females don't come to their nest very often, and so then they're not feeding their chicks very often, and the consequence is survival of chicks is lower. So that is a behavioral change in the mother, which influences fitness outcome. So with plenty, and then that's a behavioral pathway. The stress pathway is another one that there's a lot of evidence for. Mothers that are super stressed, like snowshoe hare that have been smelling lynx because Canadian scientists have been pouring lynx smells everywhere. They're super stressed and their babies are smaller and some die. So that's a fitness consequence to high predation. So that's all that non-consumptive pathway.
Okay, but the density may change in fraction.
It just doesn't continue. And we stop at this second part, right? We stop at this and here we stop at prey phenotype. Some people even go one step further and make the assumption that, look, if you've got fewer babies or you live less long, the consequence will be densities will lower. But the feedback loops don't work that way. And we don't have good evidence that shows that this leads to this, this leads to this. This leads to this, yes, this to that, yes, but not the final step. And so we should stop saying that it does. Yeah.
Just out of my curiosity, this beautiful platter that lizards live.
Yeah.
So literally, the wind turbines eliminates the main predators of this lizard.
Correct.
Is there any consequences for substituting the other predators because of elimination of the main predator, for example, snakes?
Yes. Yes. We haven't seen that. The only other predator that would be relevant there would be small mammals like sculptins, but their densities are so low, and they're also generalist predators, and so they're eating the rodents on the nice food and everything else. So we haven't noticed that the lack of raptors has given another predator an advantage. But we purposely picked this plateau because it was a simple food chain. Right? The moment I get to like another ecosystem where I'm looking at predator effects and that it's a food web with many players, I bet we're going to see reduced effects. Right? I mean, that's expected. But different pathways. But here, when you've got just so few players, the consequences are so clean and so strong, which means it can.
Okay, and how long is this data?
How long does what?
This data gathering and of the statistics, for example. How long?
Depends on which study you're talking about. The wind turbine one?
Yes.
We were out on the plateau for 3 years. And so when we did different pieces of information like density, we measured all throughout, behavior, the FIDs, we did 1 whole year, and then the stress stuff. So it took us 3 years to get all that data. Mm-hmm. All right, thank you.
Thank you.